Hi. So in previous session we have learnt about the discovery of complement and what are the different pathways of complement and what are the various molecules involved in complement pathway. And now I will take you to individual pathway, and in this session I will take you to the classical pathway. So this classical pathway is, if you remember the previous session, it is triggered by antigen antibody complex. Okay, so let's look at how it is activated. So basically this is antibody dependent. This is the only complement pathway which is activated by antigen antibody complex. Therefore classical pathway is antibody dependent activation pathway. Okay, so it is activated by various kind of antibodies. Okay, this is IgM and IgG. So IgM is very effective in inducing the complement pathway compared to the IgG. Okay, and there are several subtype of IgG which you will learn when I will take up the antibody, and not all subtype induces the or activate the complement pathway. Some of the this IgG subclass can only activate the complement pathway. Here just one more information I would like to tell, that IgM has a more potential to activate complement pathway. You will understand only one molecule of IgM is needed in order to activate the complement pathway. On another hand, in order to activate same level of this complement pathway, 1000 IgG molecule is needed. Okay, this is a one important information. Of course, early stage activation needs the C1, C2, C3 and C4. Okay. So I would like to discuss in more detail about C1. Okay. So there are here you can see that there are various C1 subtype. Okay. This is basically C1 is a macromolecular complex. It's a quite big molecule, as you can see here, and this is basically consist of C1Q and 2 unit of C1R and 2 unit of C1S. Okay, so this basically here you can see that there is a C1Q, and this C1Q there is a more resolved schematic of C1Q. Here you can see that there is a triple helix structure. It's a collagen triple helix structure. I don't know if you studied the biochemistry, this triple helix structure is present in one more very important biomolecule which we call it as a collagen. Okay. So this collagen is rich in, if you remember, this is rich in proline, and this proline is basically not present as a proline, it is present as a hydroxyproline, and for making this proline to hydroxyproline there is a need of enzyme which needs a vitamin C. Okay, probably you may remember, just I am giving you as a information. So this C1Q has this collagen like triple helix structure, and this subunit basically binds with the antibody molecule, and in antibody there is a one region which we call it as a CH2 domain. Okay. So what is CH2 domain? I will show you in subsequent slide. It is better to understand the structure of antibody so that you are able to understand the things much better. And this C1Q is basically making a complex with, here you can see that, 2 subunit of C1R and C1S. Okay. So this complex is verified by electron microscopy. Here you can see there is a on top there is a electron micrograph of this complex. Okay. Now let me show you the structure of antibody. So this is just for a quick understanding. I will again discuss this structure when I will take the antibodies. Okay. So antibody has a, so this is a one class of antibody, the structure which I am showing. This is mainly the immunoglobulin G. Okay, the structure is more resemble with immunoglobulin G. Okay, so this immunoglobulin G is having a two light chain. Here you can see there are two light chain, and it is basically consists of 214 amino acid, and there is a two heavy chain. Okay, so this heavy chain, if you see carefully, there is a CH2 region. Okay, and this CH2 region basically bind with C1Q. Okay, and this basically this light chain and heavy chain joined together by various interaction, and one of the important interaction is by disulfide linkages. Okay, here you can see that these two chains are basically linked by two disulfide linkages. Okay, and there is some antigen binding region which is present in both light chain and heavy chain. Okay, so this antigen binding region basically bind with the antigen, and then there will be some conformational changes taking place in this molecule, and this conformational change will be noticed, or that will also induce the conformational changes in C1Q molecule or subunit of C1 complex. Okay, this conformational change will be detected or sensed somehow by this C1Q, and then it will interact. Okay, so there is a effector region. This I will discuss more in when I will take up the antibody. Just for your simple understanding, you should remember there is a FAB part of antibody. FAB here you can see, which is basically binding with the antigen. Okay, and there is a FC portion, and this portion is basically playing an important role in biological activity. Just for your information, antibody molecules are heavily glycosylated. Okay, here you can see there is a CHO. So this region is heavily glycosylated, and there are several kinds of antibody. Here you can see that there are 5 kinds of antibody, that is IgG, IgD, IgE, IgA and IgM. Okay, so among these several kind, IgM is a, as I told you in previous slide, that IgM is a very strong in making this classic or activating this classical pathway. Okay, and this IgM is basically pentameric in nature. So there are, you can see that there are 5 basic unit of antibodies are joining together, and that makes IgM. Okay, and this is very strong. This has a more potential to activate the classical pathway compared to the another molecule which is IgG. Basically Ig stand for immunoglobulin, and M is a type. Okay, so this is just basic information about the antibody. Now I will talk how this classical pathway get activated. Here you can see there is a very beautiful schematic, and this is a quite self-explanatory. Okay, here C1Q basically binds with CH2 domain or CH2 portion of antibody which is bound with antigen. Okay, and this, when it is binding, then C1R get activate, and once it will be activate, there will be a autocatalytic activity, and this autocatalytic activity basically activates second C1R molecule, and eventually the both activate C1S component of C1 complex. Okay, here you can see there is a very good schematic. Second step will be, once this C1S get activated, then this cleaves the complement C4 and C2. So C4 and C2, once it will be cleaved, then this will basically C4 exposes the binding site for C2, okay, and this C4 binds the surface near C1, and C2 binds C4, okay, and forming a if you remember, the aim of activation of complement is to generate C3 convertase, okay, and here the C3 convertase is basically C4B2A, okay, and this has an enzymatic activity. So once there will be a lot of C4B2A, this will convert lot of C3 into C3A and C3B, and C3B will be coated over the microbial pathogen, okay. The third step is, which is quite obvious and quite clear, that this C3 convertase, as I explained you, hydrolyze or breaks the C3 molecule into basically it will generate the C3A and C3B, okay, and C3B will be coated over the target microbe, and then it will be readily phagocytosed. Another thing that will also generate the C5 convertase. So C3B which is interacting with C2 which is interacting with C4B2A, which has already enzymatic activity, it will interact with C3B and it will acquire another enzymatic activity, that is enzymatic activity for making C5 break, okay, and that is that enzyme will be C5 convertase. So C5 convertase, I will repeat again, the C3 convertase basically make a lot of C3B. So one feature of this C3B molecule will be coating the microbe, and then it will be phagocytosed. Another aim is to make a C5 convertase. So this C5 convertase is generated when C3 convertase, which is C4B and C2A, which is having a C3 convertase, this will interact with C3B and that will generate the C5 convertase activity, and eventually the C5 convertase will be C4B, 2A and 3B. So this C5 convertase is needed for generating lot of C5A and C5B, okay. So here you can see that there will be a generation of C5B and C5A, okay. So this the C3B component of C5 convertase bind with C5, permitting the C4B2A to cleave C5, okay. And this C5 convertase or C5B binds with C6 and initiate the formation of membrane attack complex. Here you can see that this membrane attack complex is formed, and then and this is basically supported by C9, okay, and this C9 basically make a pore in the target cells, and this C9 is basically making the membrane attack complex. So there is a lot of evidences about this membrane attack complex. Here you can see there is this is an electron micrograph, and this electron micrograph is showing a big pore in the target cell, okay, and basically there is a 17 molecules of poly C9, and this poly C9 basically makes a pore. Okay. So, this membrane attack complex, it will make a pore in cell membrane of target cell. Okay. And ion and a small molecule can freely pass through this pore, and the cell cannot maintain the osmotic stability, and eventually the cell will die. So, this is all about the classical pathway. In subsequent session, I will talk about alternative pathway and lectin pathway, and I will also talk about the disease associated with complement dysregulation. Thank you.